In this lesson, we're going to examine the role of DNA in evolution and biodiversity. Let's begin with the basic structure of DNA. So DNA is often called the blueprint of life. Its other name, if you're not into abbreviations, is deoxyribonucleic acid. There's another major nucleic acid called RNA, or ribonucleic acid, which we'll talk a little bit about as well. Now, DNA contains instructions for making proteins within a cell. So if DNA is a blueprint of life, proteins really are the major building blocks of life, and so we'll get into how they're made. But first of all, where can you find DNA? So DNA is found in the cytoplasm of prokaryotes and the nucleus of eukaryotes. If we recall, prokaryotes are organisms without a nucleus, so pro meaning before, and eukaryotes are organisms like us, whose cells have a true nucleus. Now, DNA is found in thread-like structures called chromosomes. DNA in eukaryotes can also be found in mitochondria and chloroplast. We'll get back to that later when we talk about the endosymbiotic theory, which explains how the eukaryotic cell may have evolved. So, our genes are found on the chromosomes, and if we recall, genes are basically the segments of DNA that basically code for proteins. So the genes are responsible for our traits, and you may recall from earlier grades as well, we talked about alleles, how alleles are different forms of a gene. And we will get into that a little bit more as we continue our discussion on evolution. Now, back to the structure of DNA. So DNA is a very long polymer. By that, it is made up of repeated building blocks. So in the case of proteins, they are made up of building blocks called amino acids. Nucleic acids are made of nucleotides. Now, the shape of DNA was very famously discovered, really by three scientists, so by the name of Watson and Crick, as well as Rosalind Franklin. It's an interesting story and one you should maybe Google if you're interested. We may talk about it a little bit later in class. Now, the basic structure of DNA is that it is made up of two different strands twisted together. So it is a double-stranded molecule made in the shape of what we call a double helix. So that's the sciencey term that we'll use now here at grade 11. But we can see how it's in the shape of a twisted ladder. So let's look a little bit more here at the nucleotide. So these are, again, the basic building block of a DNA molecule. So DNA is made up of a sugar. So that sugar is deoxyribose, a phosphate group, and a nitrogenous base. So the nitrogenous bases in DNA are adenine, thymine, cytosine, and guanine. Adenine and thymine always join together, and cytosine and guanine always join together. So I like to remember this using the phrase, the apple in the tree and the car in the garage. It's a good way to remember how these complementary base pairs go together. So the two strands, again, fit together somewhat like a twisted ladder, with the backbone of the DNA molecule, basically the rails of the ladder, if you will, consisting of alternating phosphates in that 5-carbon sugar deoxyribose. The nitrogenous bases are the rungs of the ladder, and they are held together by hydrogen bonds. So the basic function of DNA is to store genetic information. So parents pass their DNA on to their offspring, and the DNA will code for specific proteins that are essential for life functions. A complete sequence of DNA is called a genome. So basically, for example, our genome is made up of 23 pairs of chromosomes with over 20,000 different genes. Now, the genetic code. So this is where, again, we really can help to understand why and how DNA works. So a gene is a segment of DNA that codes for a specific trait or protein. And our genes, as we talked about in earlier grades, are responsible for things like our eye color, our hair color. They're responsible for the making the proteins that that speed up chemical reactions or enzymes and so on. So there are an untold number of functions that DNA is responsible for. Now, the basic instructions in the gene tell a cell how to make a specific protein. An organism's genetic code is a sequence of bases in their DNA, the A, C, T, and G. So each gene's code combines those 4 nitrogenous bases in different ways to make 3-letter words called codons that specifies which amino acid is needed at each step in the making of a protein. So the code in the DNA is carried to the ribosome by RNA. So basically what happens is a DNA molecule is transcribed into RNA, and that RNA acts as a messenger where it takes the message in the original DNA molecule out of the nucleus to the ribosome where proteins are assembled. And that DNA message is carried out to the nucleus again, as I said, by those RNA molecules and ribosomes as a site of protein synthesis. Now, what does this mean for evolution? Well, every organism's DNA is unique, and variation occurs within a species and between species, all due to differences in that genetic code. And it is that variation which evolution acts upon. So just a little brief discussion of how that variation occurs here to end as well. So variation in a population is caused by mutation and gene shuffling. So that gene shuffling occurs through meiosis, but also through sexual reproduction when you get a combination of 2 parents' chromosomes. So we'll talk a little bit more about that as well when we talk about sexual selection and how that works. But a mutation is sort of what I wanted to focus on right now. So a mutation is any change to the DNA or RNA of an organism. Mutations in gametes are the important ones because they can be passed on to the next generation. So the image here on the slide talks a little bit or illustrates really a few of the common ways that gene mutations can occur. And so, for example, you may have a nitrogenous base being inserted into the middle of a DNA strand. You may get a deletion, so one going missing. These ones are really important to understand. So these insertions and deletions can basically affect that whole big chain of amino acids. And then another one that's really interesting and actually quite commonplace and can actually explain some fairly significant genetic disorders like hemophilia and cystic fibrosis, as well as sickle cell anemia, is this substitution. So in a substitution mutation, basically one nitrogenous base gets substituted for another, and that will potentially affect an amino acid in a particular chain of a protein. So it is important to understand that mutations, not all mutations are harmful. Many of them are, such as the ones I just talked about through genetic disorder. Quite often they're neutral, and the reason for that is because there are 20 different amino acids that can make up a protein and 64 different codes. So you can see that there's some redundancy built into the system. And then sometimes, as we'll discover, they actually convey a positive benefit. And so the next step, now that we've reviewed DNA, will be to talk about again how basically evolution acts upon DNA through natural selection. So I would like you to, now that you've watched this video, I would like you to complete the little DNA review quiz, just so I can see where your understanding is, so we can start this discovery or discussion of natural selection tomorrow.